Margaret McFall-Ngai, I'm faculty associate at California Institute of Technology in Pasadena, California. And I've also had tenured faculty positions at the University of Hawaii in the School of Ocean and Earth Science and Technology, at the University of Wisconsin-Madison in medical microbiology and immunology, and at the University of Southern California in the Department of Biology. So go all those places. I've obviously been in this field a little while. I got my first faculty position in 1989.
Yeah, that's awesome. And then how did you get interested in research in the first place?
Oh my goodness. Well, that's a very interesting question. So I've always really liked biology. When I was in high school, sort of decided I really liked biology and chemistry. It was fun. And then when I went to college, I also liked sociology. I wanted to work to help people and so on and so forth. But Anya, the funny thing about that field was that it was very difficult to distinguish yourself. Everybody got A's. I was just like, how do you do this? So I decided that I would switch my major in college over to biology. So I got my bachelor's degree in biology and then went off to UCLA where I got my PhD. When I was getting my PhD, I worked on a fish. Worked on fishes, and this fish had luminous bacterial symbionts associated with it. And my study site was actually the central Philippines. So every spring quarter, in other words, sort of from March through May, I would go and live in the Philippines when I was a graduate student. And that was really fun. And finished off my dissertation. And I was really fascinated by the fact that to use the light produced by the bacteria, well, they use it so that they camouflage themselves from moonlight and starlight. So they come up in the water and they put out light out of their ventral surface that matches moonlight and starlight. So a predator looking up from below can't see them. They don't cast a shadow. So I was really interested in the fact that this fish would made its ventral surface transparent so that it could, beam light out into the environment to camouflage. And you know what, Anya, I thought, how does it do that? And so it turned out that the only place where that was very well understood was in the eye lens. So our eye lenses are very transparent. And so I went and did postdoctoral work at Jules Stein Eye Institute at UCLA and learned protein biochemistry and biophysics there. It was really fun. I loved learning about protein chemistry. And then I went off to Scripps Oceanography for a second postdoc where I studied enzymology. And then following that, I got my first faculty position at University of Southern California. So I really just always was sort of passionate about wanting to know something about how something works, how it's put together, and whether that it's a protein, how that protein is put together to do what it does, or whether it's an animal, how is that animal put together to do what it does. I just always had a passion for that.
Yeah, that's awesome. That's like really cool. And then I know that you just kind of talked about your research with like the luminous fish,
Uh,
but could you give me an overview of your research like now?
so what happened with the fish is that it turns out that to study baby fishes, young fish, is really difficult. And marine fish. And the reason is because they have these really complex stages. They're called larval stages, really complex larval stages that are like out in the open ocean and they're very hard to find. And I was at a meeting in Siberia, actually. It was on something called the Pacific Science Congress. And so everybody in the world who studied the Pacific Ocean was meeting on the east coast of Siberia in a town called Khabarovsk for a big meeting. And I got up there at the meeting to tell them about my work on the fishes. And there was a professor in the audience from the University of Hawaii. And I was lamenting the fact that I couldn't study how the symbiosis got started in the baby fishes. How did that work? How did they get their symbionts? And so this guy came up to me and said, there's this little squid in Hawaii that the people studying developmental biology have been using to study how squids develop from the embryo all the way up to adults. And that particular squid happens to have luminous bacteria associated with it. And it does the same thing that the fish does. In other words, it emits ventral light. It's night active out on the reefs in Hawaii, and it matches moonlight and starlight so it doesn't cast a shadow. And he said, so because they've been studying it, they have been able to get it into the laboratory, get it to lay eggs in the lab, get it to hatch in the lab. And so you could watch the animal. We have been able to talk to the scientific community about how an animal initiates a colonization with persistent bacteria. And so this particular bacterial species lives along the surfaces of epithelia. Do you know what epithelia is?
Yeah, like skin cells.
Yeah, skin cells, but I'm talking about the gut cells. In us, of course, the bacteria live along our gut epithelium. And so this bacterium that's luminous lives along the apical surfaces of epithelia in this squid. And it's amazing because there are very, very few things older in evolution than the association of bacteria with eukaryotic cells. And so, you could expect that the sorts of rules that govern how the bacteria live persistently along the apical surfaces of this animal's epithelia will be very similar to the way we keep bacteria persistently along our gut epithelia. And so we started looking at this animal, brought it into the lab, and now if you came to Caltech, you'd see that we have a bunch of females and males downstairs that lay egg. The females lay eggs, and the students and postdocs in the lab, and sometimes high school students like yourself, work on how in the world against a background. So the bacteria, you probably don't know this, but in every milliliter of seawater, there are 6 million bacteria on average, 6 million bacteria. And in Hawaii, where this animal lives, Vibrio fischeri, which is the microbe that's in the squid, in a background of a million other bacteria that they're somewhere between 100 and 1,000. So, 3 orders of magnitude fewer, 3 to 4, I mean, really almost nothing. And somehow that animal and that bacteria select one another as partners within 3 hours. So you can ask the question, how in the world does an animal recognize its partners in the background of all that noise out there? And so it's really fun because the students have been able to figure out all the steps in that process. And it turns out to be a very complicated dialogue between the host and the symbiote. Are you the right guy? Yes, I'm the right guy. Are you sure you're the right guy? Yes, I'm the right guy. And so that over time, the animal eventually gets colonized by its symbiote. And in fact, the only bacterium in the seawater that gets inside of the animal is this bacterium. Inside of the light organ is this bacterium. In other words, the selection actually occurs on the surface of the organ, and then they go in and they're persistent throughout the life of the animal.
That's so interesting.
It's fun.
It's crazy how there's only one bacteria type in like the 6 million.
Of this one bacterial type, there are probably 100 to 1,000 cells against 6 million cells, other bacterial cells. And it is amazing. And the way they do it is over evolutionary time, there's been very strong selection so that the animal only recognizes that bacterium. It makes a special conversation, a special language goes on between the two of them that the both of them understand. And it's pretty cool. It's pretty cool.
Yeah, that's awesome. And then what were some of the biggest challenges that you faced in research and how did you overcome them?
Oh, that's a good question. Federal funding is always limited. And so one of the major challenges that scientists have, I've been very lucky because I've been funded my whole career. But one of the major challenges is to compete for federal funding so that you get to do your dream, so that you get to do the research that you want to do. And so, for instance, my lab has been funded by National Institute of Allergy and Infectious Diseases for decades, out of the National Institutes of Health. And, I have to sell a squid to many of these people on the study section are physicians and stuff like that. And I have to convince them that because the language is evolutionarily old, we're liable to learn very basic principles of how interaction of bacteria with the apical surfaces of epithelium works. So that's one of the major challenges. And I think NIAID typically has a percentage of success, somewhere between 8 and 12%. So what that means is that on average, only about 1 out of every 10 scientists is successful with a given grant. And so you have to write a lot of grants. You have to, make sure that your lab stays funded. So that's one of the major challenges. Another challenge is to make sure, so you have graduate students, you have oftentimes undergrads in the lab. Sometimes, like I said, I have had lots of high school students work in the lab, and high school students, undergraduate, graduate students, postdocs working in the lab, and technicians. One of the challenges is to do your best by them. In other words, to teach them well and to recognize when they're going to be successful and help them in whatever way you can to be successful. And there are sadly times when you have to tell them that you don't think that this is the right profession for them. And so those are challenges. It's the good and the bad. So you have in science to be successful, you really have to be able to accept failure because most often you're doing something that no one's ever done before. And so you have an idea of how it might work, but you can't always be sure. And so you'll try something and it won't work. And then you'll try something else and it won't work. And you'll try something else and it won't work. And eventually, so you have to be very patient. And if you have a hard time with failure, it's hard. You just have to get used to that. That's another challenge. I have to say, this is the best profession ever. And so I feel like I've been very, very lucky to be, a professor my whole life. And the reason it's so fun is every day is different and you discover things on a regular basis that no one's ever discovered before. All that it's just such a gift. My students know I work really hard. And at one point, one of my students recently asked me, how many days of the year do you work? I sat back and for a second and I said, well, maybe 2. And they laughed and I said, they said, what do you mean you only work 2? You're working all the time. And I said, it's not work, it's fun. So I feel like it's a tremendous gift to find the profession that you're really excited about. I mean, one of the things that's been very exciting to me, challenging to me in the beginning, was that I got my first faculty position in 1989, and I was the first woman hired into the history of my section in the department. First woman in the 110-year history. When I took my second position, I was the first woman hired at the University of Hawaii in the unit in which I was a professor. It's very hard because the men are just by their nature, they're not used to having women around. If you're interested, there's a tremendous book that was written in 2023. And the name of the book is The Exceptions. And it's about women who were in science. It's mainly about the women at MIT who were in science in the 1980s, 90s, and into the 21st century and the struggles that they had. And so the other thing that you have to know yourself and you have to know what you're passionate about. And so some faculty are very, very, very good at competing. And so I make the analogy that they're like people who work on Wall Street in Manhattan in tuxedos, or in suits. They're like that, And so those guys are, they're really good at elbowing and making their way to the top by competition. The other way you can be a very successful scientist is to be a pioneer. And that is that you're out on the plains of Kansas in a set of overalls with a sickle and a shovel. And women are particularly good at being pioneers because you're not in this rat race of a competition where you'll be left out. And so many of like Barbara McClintock and other women who have won the Nobel Prize, Jennifer Doudna and whatnot, they were pioneers in their fields. And so one of the things when potential graduate students or prospective graduate students come in and interview with me, I always say to them, ask yourself which of those avenues you like. Would you like being in a very well-funded, rat race kind of, rich people, in Manhattan, or do you want to be a person who is forging the frontier? And you have to make that decision. And of course, I tell them, if you want to be in Manhattan, I'm not your advisor. And the students in my lab, Anya, they all do things that I've never done, lots of them. In other words, I can teach them what is the nature of a high quality question to ask. What will get your paper in Science, Nature, or PNAS or something like that? One of the premier journals. But most of the time they go off as pioneers into something and they make it their own. And a lot of times I don't know what they're doing. They tell me what they're doing, of course, but I didn't develop it and discover it. They did. It's a different way of being. Whereas if you go the easy route, all the methods are worked out. Everything's worked out. You walk into somebody's lab, they say, here's your project, do it. And so you know exactly what they're doing and exactly what they have to do to make it work. And so it's a very different culture.
Yeah, that's so cool. I never really thought of like science as pioneers versus like a kind of a rat race, but
Yeah.
that's really cool. And then where do you see like your research field expanding to? Because I know that there's like the rise of a lot of new AI and there's all these new like, technological advances, but like what
Mhm.
are some interesting topics that you think can be further explored within the next 5 to 10 years?
Beginning in 2006, there was a huge revolution. So you know about the human genome. It was, like 1998 to 2003 or something like that. I can't remember the exact years, but it took 2.5 billion years to sequence a single human genome. Today, you can get your genome sequenced for $600. Okay, so this is a huge change in the cost of sequencing. So one of the major fallouts of that was that in 2006, there was a huge change in the cost of sequencing DNA went from $6,000 a megabase to 2.5 cents. $6,000 to 2.5 cents by a breakthrough in technology. What that breakthrough in technology did was it opened a world that we didn't know. And that world was the world of the microbes. And so most of them are not culturable. Most of them are out there, in the environment. We don't know who the hell they are because we couldn't know who they were. But now we can sequence their genes and know who they are. And so what happened beginning in 2006 was we began, like I said, to discover a world we didn't know. And it turns out that back in 1979, it was thought that there were 7,000 species of bacteria, which is the same number of species as there are snakes, which is kind of absurd. So it turns out that there are millions and millions of species of bacteria. We don't know how many yet, but this incredible change in technology allowed something called Terra Oceans, where Craig Venter, this huge person in sequencing, went all around the world. Terra Oceans, they went all around sampling the ocean, and defining what you call the microbiome, the Earth's microbiome. In other words, looking to see what kinds of microbes are out there. And every time they would do a sequencing, they would get new organisms, new species. It was just breathtaking. They did Terra Oceans. They did the Earth's Microbiome Project, was a lot of terrestrial stuff in the soil, in the trees, in the animals, in the whatever. And then they did the 10-year Human Microbiome Project. And it turns out that about 50% of your cells are microbial. 8 of the 10 organ systems that you have, or 9 of the 10 organ systems that you have components that associate with bacteria and other microorganisms. I'm very bacteria oriented, but other microorganisms too. It turns out that the microbial world is the basis for the health of all corners of the biosphere. And so we just started to recognize this less than 20 years ago. And it's truly remarkable. The largest change in our view of the biosphere since Charles Darwin in the 1850s. We now know that the vast majority of the diversity is invested in the microbial world. And like I said, it's responsible for the health. Its balance is critical in the soil, in the oceans, in our gut, and so on. So I think for the next many years, that would be one major area that we would continue to delve into and understand better. One of the things that I think absolutely has to happen, and I'm teaching a course at Caltech with a microbiologist. I'm an animal biologist, but I'm teaching a course with a microbiologist next semester because I feel very deeply and strongly that there needs to be an integration of microbiology and macrobiology. The field of microbiology has been separated from the field of plant and animal. And plant and animal biology has been working on everything from populations and so on and so forth, all the way down to cell and molecular within animals and plants, right? And then over here is the microbial world that actually, it turns out, invented everything, something we're learning, invented everything. That has been studied separately. So people will have gone through biology, through microbiology, without having any animal and plant biology, and the animal and plant biologists would never have any microbiology. There are still many, 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 many universities in the country that the biology majors can walk out, have a bachelor's degree in biology with a single lecture on the microbial world in their freshman year. And that can't be anymore. That can't be anymore. So that's another place where there has to be really tremendous change. I think that AI will be something, actually, it's very interesting because at Caltech, I would say 50% of the undergraduates here are computer scientists. That's the worst thing to ever go into at this point, is computer science, because I use AI every day. This morning, into Google, I typed in optic nerve actin, and it came up with just like that. It says, I'm not kidding, it was like in 3 seconds. It said, the optic nerve has high quantities of actin, and it's in the astrocytes and in the neurites, and it gave me 3 or 4 references to go to, and so I think that a lot of people who are in various fields are afraid of AI. I'm not one of them, because I think that pioneering, AI doesn't even know it. Those data aren't there yet. It's a funny thing, when I was at the University of Wisconsin-Madison, I taught the introductory biology course to the honor students. So I'm teaching the honor students. And so I gave them an assignment. And the assignment was go to the CDC and see what the list of pathogens are, the bacterial pathogens like Vibrio cholera, and so on and so forth. And I gave each of them, there were 20 in the class, and I gave each of them a different microbe that was in the database back at the Center for Disease Control. And I said, what I'd like you to do is I'd like you to go to the database on the human microbiome that's now out there. And I'd like you to find out how many of these that are pathogens are actually relatives, in other words, congeners in the same genus as members of your microbiota. it was so funny, Anya, because they came back to me and they said, there's no information on that. And I said, you're making the information on that. You're the one doing this initial study to ask how common it is. So they didn't have time to do anything more than that. But there was somebody who visited, who's a very famous researcher who visited, and she studies Neisseria gonorrhea and Neisseria meningitidis, which cause gonorrhea and meningitis, right? These two bacteria. But Anya, there are 12 Neisseria species that are related to those two that are part of your normal microbiota. There's one in your mouth, there's one in your gut, there's one in your nose, there's one, on your skin, so on and so forth. So these Neisseria species, I said to her when she finished her talk about how Neisseria meningitidis that causes meningitis, she was giving a talk about what it does to the host. So I raised my hand at the end and I said, so how much of that conversation, how much of that molecular conversation is carried out by the members of the normal microbiota? In other words, to what extent are meningitis and gonorrhea spies on a pre-existing conversation that animal host has with its normal Neisseria species? And she looked at me and she said, I've never thought of that. To her credit, she went back to her lab. She's from Arizona. She went back to her lab in Arizona and she did a phylogenetic tree. What a phylogenetic tree is it shows the relatedness of a bunch of animals or a bunch of bacteria or whatever. So if you did a tree of humans, there would be a tree that had us most closely related to, chimpanzees, and then the outside of that would be orangutans, and outside of that would be, some of the lower primates. But what she did was she made a tree, and the animals or whatever at the end of the branches, those are the most recently evolved. And so what she did was she did all 15 or whatever it was Neisseria species, and lo and behold, Neisseria meningitidis and gonorrhea had evolved out of the normal microbiota. They were younger than the normal microbiota. And so, it just goes to show that, we just have to change the way we think about things.
Yeah, that's so cool. But like that's all the time that we have for today. And thank you
Okay.
so much for meeting with me.